Good afternoon, everyone. Could you hear me? Okay. So I'm Mathieu Bourgey. I am Bioinformatics Manager at the Canadian Center for Computational Genomics, which is the genomic platform, the bioinformatics platform in McGill University in Montreal. And today I will talk about genome alignment, and tomorrow about variant calling. So as you may know, this is Creative Commons content, so you can use it, you can reuse it, you can modify it. At the moment, you give the authorship to the original persons. So before we start, what we'll look during this lecture and the practical. So we'll introduce NGS technology. So you already have a big introduction this morning done by Jared. So I will really go fast on that point. I will mainly focus on Illumina approach because it's what we use for this, usually doing DNA alignment and DNA analysis. The idea is to understand the characteristics of the data and to understand for each type of the data what are the problems that this data bring when we talk about alignment of this data to reference genomes, to identify what are the possible sources of error and how we can correct this type of error or take them into account. We'll also learn about the different terminologies, the different file format, and at the end, in the practical, we'll do the first step of a DNA-seq pipeline. Okay, so technologies. So NGS start many years ago with a main genome project where sequencing was really long and expensive, and it was a revolution when we start to build this new technology where sequencing become cheap and fast. So it's an extension of what I've been done with at the beginning at the clone level, where at the clone level the main problem was that when you were running an analysis, you only analyzed 96 seconds, and it costs a lot of money. Now, what we use the cluster approach. What that means, so for an Illumina machine, all the same approach that you have for clones, you have it, but you have millions, this time for one run, you have millions of time the sequencing on one single run. So you can generate up to 500 million of sequences at the same time in the most recent machines. So how it works? For Illumina, it's called sequencing by synthesis. The main idea of that technology is you take your DNA, you cut it in a fragment where you approximately know the size of the fragment you generate. You already talked about that in the previous lecture. So you decide one fragment you want. On this fragment, you add some adapter at the two ends of the fragment. You load this fragment on your flow cell. In your flow cell, you have corresponding probe for the adapters. So the molecule will come and fix to this adapter. So you will have all this DNA string attached to your flow cell, to the surface of your flow cells. So your flow cell will not be empty. So where the magic of the synthesis of the technology is when we do what we call bridge amplification. So you will have your string of DNA, but they will still have on the flow cell some free probes. So your probes will match another probes which have the complementary of the second end, and that will create a bridge between the two probes. And then you will do PCR amplification of your fragments. And then you will have another copy of your data in close proximity in your flow cell. And then you will denature the double-strand molecules to have single molecules, and you will repeat this bridge amplification again and again. And at the end, what you will have a cluster of sequences that are closely physically located, that represent the same initial DNA fragment. So when this cluster has been generated, what you will do, you will start your sequencing. So you will have an adapter that is that fix the top of your sequence. And then after the adapter, you will bring the different, you will bring your bases, and each cycle of the machine will bring one basis to each sequence. So you can imagine that as all of the sequences from the same cluster are representing the same DNA molecule, they will bring the same basis for this cluster. So each basis is linked with a specific fluorescent. So after each cycle of sequencing, the machine will take a picture of the flow cell, and for each cluster, one dot of a specific color will appear on the image, and the sequencer will be able to tell you, okay, for this position, we know based on the color that this base have been integrated in the sequence. So each cycle, you will know for each cluster which bases. So if we take the first one, you see first cycle G, and C's, and T's, and G's, and A, and so on. And that's how the system works. So the four major players, I think you've done that. I will skip that part. What is important to know, based on what Jared presented you this morning, is that you have several applications, several type of sequencing. But what is important is to understand what all these machines and technology are used for. So if we go back there, there's three classes of sequencing. There's the technologies that generate small reads, which are around 100 or 200 base pair long for each end. There are medium, IC 454, which generate around 5 to 700 sequence long for each read. And there are the long reads, like PacBio or Oxford Nanopod, which generate large sequences that could go up to 50, 80 kb read long. So what is important is to understand each technology, each type of technology, what they are being used for. So the medium sequencing is usually used to do small de novo genome sequencing, but not so much now that we have a really efficient long reads. But it's mostly used to do amplicon sequencing, to do validation, and to do metagenomics. The short reads are those that are the most used, and they are used for everything that is usually a reference-based approach. So you know your reference, you know your assembly, and you will do like whole exome, you will do transcriptome, you will do DNAs, you will do ataxic, you will do IC, all this kind of approach where you know the reference and you don't need to reconstruct your reference. You can use with short sequencing because they produce more sequence, they are cheaper, so it's where people are using. The long reads are really used for genome assemblies most of the time. They are also used to do haplotype sequencing, to do targeted sequencing, to do some epigenomics, also sometimes some metagenomics where we want to cover the full 16S region. So all this data could be used. I had another technology here that is not a sequencing technology, it's more a library prep technology that is really becoming a new player in the field, which is called 10x Genomics. And it's a way to generate your library. Based on a bead system where each fragment or each cell will be amplified and the library will be generated around the beads where you will be able to add some specific barcodes. And this technology is really now emerging and efficient to do single cell analysis, to do de novo genomic sequencing because they are able to generate artificial long reads, and also for whole genome sequencing because they allow you to do some long haplotyping of your molecules. So it's really a new technology that is really becoming more and more used at this time. Yes.
What was the middle column?
Here?
Yeah.
It's just my shameless advertisement. It's the number of sequencers we have in my center. Just to let you know why I know my expertise because what we use, and if you want to do that, just let me know. I can make contact with your center. But it's the number of what we have here at McGill University Quebec Innovation Center.
Sorry, the 10x Genomics, you said it's not a sequencing method?
No, it's a library prep method. So you generate your library prep. At the end, you take once your library is ready, you put it on Illumina either NovaSeq or on iSeq 4000, and you will have short reads, and then in your reads, one of your reads will contain all the barcode information that you will use then to separate your reads between the different cells, if you do single cell analysis, or different haplotypes, or single molecules if you do classical DNA.
So it's a preliminary step.
Yeah.
Okay.
Yes.
Right.
Okay. Last thing I want to say about NGS technologies is most of people come to me at McGill to ask me how can I design my experiment, this is my project, what should I use for sequencing, what should I use for analysis, and why I put this slide is because a lot of people arrive and say, okay, I've got this money, I've got this time, and I've got this question, what should I do? And this is one of the main messages I want you to have is this is the right way to work. When you design your experiment, you first need to think your question. Based on that question, what read length I need to use, what library type I need to use. Based on that, I will choose a sequencer, technologies, what these technologies will have in terms of error profile, because if I'm doing like, if I'm interested in indels and I'm taking some technologies that is more prone from indels, I will have a bias in that, I will have more trouble to do analysis. So it's more that kind of question you need to first think for designing your experiment, more than cost and turnaround time. And after, when you think about cost and turnaround time, you will see, okay, for that technology, what are my potential barcoding? So can I multiplex my data and try to save on cost? But if you drive your experiment design based on cost and turnaround time, you will probably don't be able to have the best answer on your question. Okay, so now let's go on what we can do with the data. Just to give you an idea of what we'll see during today and tomorrow, this is the workflow of analysis that we will do. Today we will only cover this part, so approximately half of the workflow, knowing that the other workflows are more complicated, so it's what we split in 3 parts. So if I summarize this part in a more simplest way, what we want to do, we want to start from FASTQ file, which are the type of format you will receive from your sequencer. We'll see what that means after that. And what you want to do, you want to generate alignment of high quality that you will be able to use, so alignment files that you will be able to use for the next step doing your variant coding. So what we will cover today. So what is the FASTQ file? So the FASTQ file is what your sequencing center will provide to you. So it's not directly what goes out of the machine. What goes out of the machine, and especially Illumina machine, is BCL file or QBCL. So we usually do some raw data processing to go from this BCL to FASTQ file. And for each sample you have sent to the center, you will receive either 1 file or 2 files for this center. 1 file if you have take your molecules and you have do only sequencing from one end and stop, or you will have 2 files if you have do a paired end sequencing where you start the sequencing from one end and from the other end. So both files will be similar in terms of format, which is represented here at the bottom of the slide. The format, so for each sequence you will, and in each file you will have 4 lines. The first one, the first line represents the ID of the sequence, so the names that have been given when we generate the sequence. So usually it's go with some tricky name which usually link to the sequencer name. Then you have some positional information about where was the cluster of the flow cells. Then you've got the second line, which represents the sequence that have been generated. You have a third line that can be used to add a copy of the header here, but the beginning is different. So why we have this third line is just because at the beginning, the first sequencer were separating these 2 lines and these 2 second lines, so they need to have 2 headers. So when people decide to merge all the FASTQ file in one in one format, they keep the two header, and some technologies still give a second header, but Illumina will never give you a second header. They will just give you a plus sign on the third line. And what is important and a bit more complicated to understand is the last line, the fourth line. The fourth line is the quality of your sequence. So it means that each symbol represents a quality that represents each basis of the second line. So why we don't use a direct number? Because quality is measured as a matrix, so it's a number. But we cannot use a direct number because, for example, if my first basis have a quality of 41 and the second basis have a quality of 3, if I use number, I will have 4, 1, 3. So I know if 4 represents the first base, 41 the first base, 413 the first basis. So it's complicated if you use numbers. So the concept that people have used for that is to use ASCII characters, because ASCII is an informatic format you can convert into numbers. So each character, each symbol, using an ASCII table can be converted to a number. So you are able to say, okay, my first character here, 4, is the value of the quality is not 4. The ASCII value of 4 is the value of your data. So when I'm talking about value, I'm talking about the base quality. So this is the value that are given, and the value are what we call a Phred score. A Phred score, you will see almost all the quality you will see in genomics are Phred score. What that means, a Phred score? A Phred score means this value here represents -10 log base 10 of a probability. So all the quality you will see, mapping quality, base quality, variant quality will be a Phred score. So this representation. The only difference between all these values are what is the probability that is represented in my formula. In terms of, so the quality that is represented for each type of a matrix is a probability to have an error. So in terms of base quality, the probability here is a probability that the base that have been called have been called wrongly. So if I've got a base quality of 30, I mean I've got 0.1% chance that the base that the sequencer called was wrong.
So you start with the ASCII and then you go to the number, and then from the number you go to the probability?
Yeah, so usually we use a number. It's just to give you an idea of the probabilities. We don't use directly the probabilities. Some algorithm will use it to do some variant codings, but most of the time all the tools will directly use the base quality, not the probability. But it's just to give you an idea of what that means, because if you need to use it, you need to know what type of error it represents.
Sorry, that fourth line is the ASCII value of the average for that sequence?
No.
Or
For the read, so for the cluster that have been seen. So from the cluster, one base has been called for each cycle, and it's the quality of that base.
So that's all in the fourth line?
Yeah. For one sequence, so for one cluster. So, yes. Go, start.
So now, what's the best quality scores from this chart? I mean,
it depends of the sequencer. Each sequencer measure as a way to measure is quality is error there. For Illumina, now I think the top is around 40. 40 or 50, I don't remember. But they have changed now the last version of the Illumina. If you take the NovaSeq, they only use 4 levels of base quality. They break it in only 4 categories, so it's not a continuous value that we have as previously. So what we do from the FastQ, usually what we do, the best thing to do is to do QC of your data. Another message you need to have and to remain from my talk is matrix, matrix, matrix. Looking at QC is really important to understand your data and to be able to assess the quality of your data. So one of the first QC you can do on your FastQ is based on this base quality. So is to generate this kind of graph. So what this graph represents, you see each yellow boxes represent a cycle of the sequencer. And what is in the yellow box is the distribution of the base quality at the first, for the first cycle, so for the first base of each read. So you have the full distribution. So you know that all your reads at the first bases have a base quality which is higher than 25 for this example, and you have the distribution. And it's a common Illumina pattern of IC Illumina pattern. What you see is that in the first cycle you have a good quality, but the machine is still doing some calibration of his laser, so the quality will increase during the first set of cycles, and you will reach really the top of your qualities. And then you will see that your quality will decrease along with your cycle, which means over the time when you add bases, the quality of your cluster will diminish, the quality of the sequencing. Why is that? It's because there's a phenomenon that we call phasing in your cluster, which means that all the base, all the sequence in your cluster will not integrate the next bases at the same time. So there will be a small delay between the different sequence in your cluster. So in the first cycle, the difference is so small that it does not make a difference for the cluster. The dot is always a big dot from the same color. But while the sequences go over, these delays can have some sequences that are one base in advance, some sequences that are one base late, if you compare to the majority of the sequence in the cluster, and that will create some other colors that pop up in the middle of the cluster, and that will decrease the quality of the dot that is read by the sequencer, and it's why the quality will decrease. So that means when the picture will be read, when the image of the flow cell will be read by the sequencer, will say, oh, it's yellow, but kind of not a pure yellow, because there's other bases that start to pop up with other color in the pool of sequence in your cluster. So it's what explains this phenomenon. If you have this kind of graph and you see in the middle like a big decrease in your quality, usually that means that you've got a problem during the sequencing. But this phenomenon is normal to a certain extent. If it's got too low, there's an issue. But this is a phenomenon. This is a normal phenomenon. So why we look at that is because we want to ensure that the quality of our sequence is good. Because if you have low quality, it will have an impact on what you will do later. So it will have an impact on your variant calling. Yes?
Is this something that's provided to you with your sequences, or is it something you generate for yourself when you receive that?
It depends on the center. Us, we provide them, but there's tools to do that. We will generate, I will show you, because we'll start from the FastQ, I will show you a tool that you can use to do that, but there's many tools that you can use, like FastQC, to be able to generate this kind of graph. Yes?
Can you do this for the code generation? Or is it just for QC?
You can do this one. You can do for everything that have quality associated. Every FastQ.
Can you do this for pipeline?
Yes, because SpecBio has a quality, so you can generate. But if I remember, the quality is not depending on which version of PacBio. If you use RS2, or if you use SQL, it's not the same format, but probably you will have to do kind of file manipulation to generate a FastQ, and then you will be able to. No, it's just that, for example, the new FastQ, the new PacBio SQL provide data in BAM format. The old one provide data in FASTA and in movies. So you need to do some kind of data manipulation to generate FastQ file, because most of these tools take in as input a FastQ file. So it's the same information, but that is displayed in a different format. So you have converted to switch from one to the other. Yeah. So if you have a BAM file, you can just use like PK or GTK to do SAM to FastQ, or I think also some tools are able to do that. So to go from SAM to your FastQ, and you can generate the graph.
I was looking at this as a
they should have it. Another QC you can do on your data, and we talked previously during module, is to look at the positional base content, which means for each cycle, like the same type of graphs than previously, you will have 4 lines that will represent the percent of bases in all the read for this cycle. So you expect, if you are working with exomes, that you have 50, so 50% of GC, 50% of AT, so 25% of each basis. If you work in whole genome, you will expect to have 60, 30, 30, and 20, 20, because we have 60% of GC. So depending on what experiments you will see, you will expect to see the different type of graph. Here, what you see, it's RNA pattern that we saw. It's clearly the first base, because when you do RNA shearing, the method to do when you do RNA shearing with Nextera libraries, it's not a fully random pattern, so it's why we saw this non-random pattern. But after that, you see that it's come from a flat line at 25, because we're on the exome part of the genome.
So the first pattern is like how you catch the RNA?
Yeah.
Oh, okay.
So there, just to comment, some pipeline remove that, some other do not remove that, because it's not a random, it's a no random pattern, but it's still genomic sequence. So there's a kind of debate: should we keep it or not? Us, we usually keep it. Another type of QC is to look about known sequence. So when you do your sequencing, you do sequence engineering. So you add some adapters, you add some for library, for sequencing. So all these sequences are known and are not genomic sequence. So what we can do is we can look into our reads and see, do we find this sequence? You should not find this sequence if everything works well. Sometimes we find it, we will see why. But so one of the QCs is to ensure that there's not so much known sequence or engineering sequence that you see in your data. Another type of sequence you can do, not so many people do it before alignment, is to look at the duplicates. So you know how many reads are duplicates. So duplicates is how many times you've got a copy of the same initial fragment in your library. The best is to have almost no duplicates. You want that every DNA fragment you put in your library generate one single reads. You don't want to have like 10 reads that represent the same initial fragment, because that means you just overcount the same biological fragment. So it's a good matrix. The less you have, the better it is.
Or even like cancer data that duplication.
No, because we don't talk about duplication of biological duplication. It's technical duplication.
But it can manifest here. Like if you have a piece in the genome that's duplicated, you will get it. Right?
No, because what we count is really the same read of the same fragment. As your reads have been randomly placed from your DNA fragment, you will have the same. And when you do high coverage, you have several copies of the same region, but the reads are not the same initial DNA fragments that have been cut and put in the libraries. It's really you cut, so you have several copies of your DNA, and you cut it in pieces. So if you have duplication, you will have more pieces of the same region,
right.
But you don't want that one pieces generate several reads.
Okay.
Sorry, why does that happen?
Well, I will explain it later. Another type of QC you can do, and what we do at the center, is we take a given number of reads. In that case, it was around 100,000 reads, and we blast the reads, just to ensure that what we have put on the lane is what is expected. So if you have put, you think you put human DNA on your data, and you've got this pattern, you say, oh, there's probably a mix-up or something went wrong at the center. So it's just a way to have a QC of the data. So now that we have understand what is the FASTQ and how we can look at them, the next thing we do, usually we do trimming. So what is trimming? Is the way to filter the read before alignment. So filter either remove some read or remove part of the reads that are of bad quality. So the first thing we do when we do trimming is to remove adapters. So I say there could be some known sequence. If you have your read here, you have the linear fragment you have selected, and you have the two adapters that have been added for allow the sequencing. When you do your sharing, you do your sharing targeting a specific insert size. If your insert size is small, or if the standard deviation, what you get from your sharing has a large variability, you will have most of your fragment on the side you select, but it's a Gaussian distribution, Gaussian normal distribution. So you will have some fragment from the tail, and if the tail is too large, it could really reduce the size of the fragment you are catching. So if your fragment here is lower than the read length you will generate, you will read your reads, and at the end, when your fragment is finished, you will continue to sequence, but you will sequence the adapters. It's how we get this data in your sequence, in your FASTQ file. So what the program will do, what the trimming will do, is we know what are these sequences. So they will look for these sequences in your reads, and if they found it, they will cut this part because it's not a genomic part of your data. After that, the second filtering we do, we start from the end of the reads where we see there's most chance of having bad quality bases, and for each read we look at the quality. We give him a value, so the user can choose a value. Usually we choose a value of 30, so 1%, 0.1% chance of error, and we remove all the bases that are under this value in terms of base quality. So here, I think the value is there. It's a purple line here. So all the bases here will be cut. Then you go to the next basis. Is still some bases lower than 30? Yes, I cut it. And so on, until you don't have anything to cut. Then if you don't have anything to cut, you stop. After that, so you have removed part of your reads there, you have removed part of your reads there. What we do, we filter on length of the sequence. So after removing all of this sequence, if the read is shorter than 32 base pair, we drop the read because the information is really too short and we won't be able to have a good alignment for that.
Question?
Yes.
Where does the 32 come from? Is that like an arbitrary number?
Yeah, it's an arbitrary number that we use here in our facilities, but it's a number that has been commonly used by many others. It's just like under 35, 30, you have too much. The read usually it's really hard to map it with confidence. So the read will just create more noise.
So it's a commonly accepted
Yeah.
number.
But some are, as I said, when you work on NGS and when you analyze and process your data, there's one common thing you need to understand is that every time you do something, you make a choice. And you have to pay for this choice. So it's why I'm really focusing on that to make you understand all the concepts behind the analysis, because when you will do your analysis, you will have to do some choice, and you will have to understand what the consequence is. So I know that some groups prefer to be more stringent because they want really high quality, and they will cut at 50 or 75 for a read length of 100 because they only want really long read and quality. But that means you will lose some coverage. And coverage is also important when you do variant coding. So each time you decide, it's a kind of balance. I want to minimize my cost. I want to maximize my benefits. So, and 32 is the one that is commonly accepted value. What is important when we do trimming is the order on how we do that. So you need to do first removing adapters and then trimming for quality. And some software, it depends how you pass arguments, the order of how they will work. So because you don't want to remove quality first and then to look for adapters, because if you have removed your read, part of your read there, you reduce the sequence, the red sequence here, and you maybe it will be more difficult for the adapter to see if that red sequence is really an adapter. So you prefer to maximize the sequence to look for the adapter, to have the larger adapters, so the more to increase the chance of recognizing the adapters, and then you remove the quality. So to do the trimming, today we'll use Informatics, but there's way other tools that you can use. So choose the one you're used to, or the one you prefer, the one that corresponds. So all of them have some kind of different features, different way to work. So you can go read the manual of the different tool and choose the one you prefer. So now we have done trimming. So we have the best quality reads we can have. The next step is to do the alignment. So the alignment,
Sorry, just going back to the trimming tools, do any of them work on different algorithms, or are they all the same?
So there's different algorithm for the adapter recognition. Could be different. So it's because it's an alignment. It's an alignment approach. So there's different algorithm, and also there's some different feature. Some of the tool can allow you to do to look for the quality of the two ends of your reads. So it's more question of features, because mainly adapters, you will see, is not such a big issue,
right,
because now a lot of aligner support the fact to have adapters.
But given a same sample pool and quality threshold, they'll pull out the same thing.
It should, yes.
Okay.
Okay, so yeah, alignment. So when we do alignment, the main idea of doing the alignment is to find the best location of your read on your reference genomes. So Jared presents you assembly and alignment, and we'll talk about assembly tomorrow. So when you have your read, if you have a good reference genome, what you want to do, as I say, is to find the best location of all these reads on your genomes. Having a good reference genome is a really, really important step. If you're not sure about your reference, you could do your alignment and then look at your matrix to ensure that the alignment is good. But if you see that your alignment is not good, it's really better to do some assembly and generate a good quality alignment. Because otherwise, you will generate, if you don't have a good quality, reference, if you don't have a good quality reference, you will generate so much noise, and so much noise will be so much false positives and artifact in your variant coding that you won't be able to use this data. So really important to have a good reference. When I say alignment or mapping is to find the best location, why I don't say it's to find the real location of your read, it's just because when you use a reference, the reference sequence represents one version of a human genomes, which come from the combination of several samples that have been mixed, and so it do not represent the real genome of your sample, and this reference is not perfect. Some parts are missing, some parts are wrong, that could influence the location of your read. So in most of the case, the best location will correspond to the true location, but sometimes in tricky region, the best location is not the real location on your read on this real own genome. So it's why I say mapping is the way to find the best location. So when we do mapping, so we try to position or read on the reference genomes, and it's a big challenge. It's a big effort. Why? First, because we need to map millions of short reads at the same time on a genome that is a billion of letters. So just in terms of computational resource, it's super complicated and super demanding in terms of resource. So it's challenging, but now if you have access to high performance computing or cloud, it's doable. What is complicated what is challenging in terms of the process to do that is many locations could be possible from one read. So with the same probabilities. So sometimes you won't be able to know what is the exact location of your read. Another challenge is that you don't want to be super stringent when you do alignment. Because if you say, okay, I want to align my read, but I want perfect alignment, So doing your sequencing makes no sense because you can just take the reference genome and you will have it. You know what I mean? What you want to do is to find in your specific sample or in your set of specific sample, what are the characteristics of these samples? Got something that popped up here. Just okay. So you want to allow the individual variation to be included in your alignment, to be able to compare this variation with the reference or this variation between your samples. So you don't want to have perfect matching. You need to, so when you accept to don't have perfect matching, you will let sequencing error to be included in your data, but you also let the biological variant to be included in your data. So you will have, so it's more complicated for the algorithm to take it, and it's more complicated at the end for the step that will become for the variant coding. So there's many algorithms. So most of people know about BLAST, which is an alignment tool. Forget using BLAST for your reads. BLAST is a good tool, but it's really slow, and it's more designed for larger sequence. If you run Illumina ISeq data with BLAST, you can launch your job and come back in 3 months, and probably it will be finished, but not sure. So people have developed other type of algorithm, and the most used one is the Bureau-Wheeler transform algorithm, which is the one that we'll use, and which is the one which is included in BWA, which is Bureau-Wheeler aligner. So BWA is recognized one of the best aligner, but other can do a really good job. Novalign also is a really good one. Bowtie, GEM, all are good aligner. And be careful, here we talk about DNA, but if you start to use RNA, you don't use the same aligner because all the aligner have their specificities, and aligning read from genomic DNA or from RNA is completely different task.
When you say a good reference, is there a way to know which reference is good or not?
Yeah, if you align your read to a reference, regarding of the percent of alignment, the percentage of your read that are aligned on the reference with high quality will give you a good idea. And we'll see in the practical. We'll look at the metrics, and there's some numbers you can use. That my number I use, but it's not defining numbers. But I expect for a good reference to have around more than 90% of my reads mapped to my reference, and the higher is the better. So for human genomes, usually I end up with around. So after trimming, so when I count the read from the read that I give to the aligner to the read align with high quality, I've got around between 95 and 98% of my reads that are aligned.
But do you know beforehand how good quality the read sequence is, the reference sequence? Like if I've got a hippopotamus sample and
yeah, you can look at how the contiguity. So there's some metrics you can look when you look at assembly metrics of your reference to understand. So you can look at N50. So there's some metrics, but Jared, will you talk about assembly metrics in your?
Assembly metrics, yes, I'll definitely go there.
Okay, so you will see in Jared talk more detail about how you assess the quality of your assembly. So there's some metrics you can also look at the project, because usually these big projects they have their website, and you can see how far the project is in the development and how it's quality. So when we do aligner, what is really important is to use and to add the RG tag. RG means read group. So there's a read group tab. So what that means, so when we have alignment, if you do your libraries or the different libraries, each time it goes to the sequencer and it generates a set of FASTQ. You need to align this FASTQ independently and add a specific tag for this experiment. Why? First, because it will speed up your process. If you have several datasets, if you merge everything together and you put that to the aligner, it will take more time to the aligner. If you keep it split for the alignment, which is a long task, it will parallelize your task naturally and it will go faster. Second is, and the most important, is to track where you will come from. Because you will do your alignment by read set you will have. So each read set is one experiment from the sequencer. So you take all your read set, you align them, then you merge your read set, and then you start to look at your variants, or to look at specific patterns, structural variants, or whatever, and you start to see something unusual. So what you will do, you will take your BAM, look in IGV, and say, okay, split my reads for this region where I see this weird pattern, split my reads per read group. And if you see the pattern is present in every read group, that means it was present at the beginning in every samples, so it's probably a biological effect that you see on your reads. If you see that only one of your experiments shows this weird pattern and not the other, that means that it's probably a technical issue that you're facing in this experiment. So it's really important to each experiment to keep a tag that you're able to use to track back where the read comes from. Also, you will, yeah.
So that only pertains to the sequencers that have multiple lanes, right? Like in MySeq, that wouldn't pertain to.
Yeah, so each time you sequence on multiple lanes, oh no, it could be a MySeq, but if you do 2 or 3 runs of MySeq for the same sample, each time you do a run of MySeq, you generate one. You have one output of MySeq, you align it, and you put a specific read tags. But it could be, yes, it's usually more used by lanes, because on MySeq usually you don't do so much redo or complementary runs.
I thought that the lane number was embedded in the FASTQ file.
In the read name, but if you have the same run numbers that use different lane, so you don't have it, you won't be able to retrieve it. And especially IGV or others too won't be able to want to look at every read to look in the read name. So when you do your alignment, what you end up with is an alignment file, which is either a SAM file or a BAM file. So SAM is a text format, so most of the time you won't use it because it's so big that you won't use it as a text. You will use the binary version, which is a compressed version. As you say, each file could be between 10 gigabyte to 500 gigabyte for each BAM, depending on what you are sequencing, what depth of coverage. So. And what will be the format of the SAM file? You will have one line per read. I mean read 1 or read 2, so one line for the read 1, one line for the read 2, which are divided in that format. So the first column will be the read name. Then there is a flag, which tell you, gives a kind of summary of how the read have been mapped. So we will see during the practical what that means and how we can use it. Then you've got the reference position of your reads, so the chromosome and the location on this chromosome. You've got the mapping quality, the thread score of your reads. For your reads, you've got the cigar string. The cigar string gives you an information of how the sequence pair base have been mapped. So here it's a 76 base sequence, and they say it's 76 match. So it could be like 20M, 1B, 4M, which will make 20 match, 1 deletion, 4 match. So it tells you how your sequence in terms of bases have been matched. So this one is more tell you technical information. So is it match or unmatch? Is it aligned, unaligned? How is the other aligned correctly in the same direction? So this is more general information, and this is really the base in the read information. What is important to know is that match for an aligner don't mean it's a perfect match. It could be a substitution. So match, it means that it's saying that this base corresponds to this region. Whatever is it, it's the exact same basis. Then you've got the information of the other of the mate, so of the read if you are in the paired modes. So the equal sign means that the mate have been mapped in the same chromosome, and you've got the coordinate, the origin of the mate alignment, and you've got the intersize. Then you've got the sequence in bases. Then you've got the base quality. And after that, you will have extra flags that are used. I'm not describing this flag here because every aligner uses his own flags. So we generate, so we have a fast view, we do the trimming, we align the trimming, so we generate the alignment. So what we need to do next is to refine our alignment. Yes?
Can you quickly about and explain the concept of main position? The main position.
Yes. So you've got your sequence, you've got your read. So your read will be there. For example, you could do, oh, I got. Okay, so I will have here 4 matches. Here I've got 1 deletion, and here 2 matches. Here is how the read have been positioned compared to the sequence. But that don't mean that here I could have A, B, C, C, G, T, G, T, C. But my read could be like that. In that cases, he had put his as a, he says the position is correct, but it's not a match in terms of this is not the same basis. So he tolerates to have substitution in the match. So it means that he's saying the right location is there, but he don't say that it is the exact same basis than in the reference.
How does it know it's the right location though, if it's not the same base?
Sorry?
How does it know it's the right location?
Look at using all the other bases. It's kind of
Oh,
a seed, extension of the seeds,
Okay.
and so on. So just because if he, for example, if he removes that, if you change that, or if you include an insertion of deletion, it will create too much mismatch that it say, okay, no, I need to put this base here to have the continuity of the rest of the sequence. But this is not the exact same base, but this is substitution, and I'm tolerating this substitution. Yes?
Your cigar score, is that an average of your base qualities?
No. It's just a description of the position of each base on the sequence. But positioning the read before, the process of positioning the reads is based on the base quality and how we'll do that. But here, it just when you have finished the process of the different positioning or different it try to position, say, okay, this is my best position I can find, which is based on base quality or mapping quality. Then I will describe that as this cigar string. And the quality of the mapping will be described by the mapping quality of your reads.
So what's the scale for that?
For the cigar string or for the... What do you mean by the scale?
Like, what's the worst score you can get and the best?
It's not a score, it's just a description.
Okay.
As you say here, this could be a cigar string. They tell you my sequence is 4 match, 1 deletion, 2 matches.
Right.
So you could have some cigar strings that are like that, because you could have soft clip, hard clip. You could have a lot of events that could arrive in your sequence when you position your sequence on your reference. So it could have deletion, insertion that can be had. So the worst would have like few match, deletion, few match, deletion, insertion, few match. So
I was referring more to that 60. Right?
The best quality, the mapping quality. So the mapping quality, the worst is 0. That means I cannot map.
Okay. Sorry, the arrow seemed to point to that number.
Yeah, sorry.
The first base here is an aim up there?
Yep.
And the 60 is the best?
Depending on each aligner is maximum. So 60 for BWA is the best. For star, for example, if you do RNA, it's 255.
76 and 76 matched.
Yes. Yeah.
The first base pair is an N?
Yeah. It probably means that when when the sequencer has been run, it didn't arrive to a quality base. So I put it there. And as we do the trimming from the end, if the quality was going up there, then look at that. So it's why the end stayed the sequence.
Is it a global alignment or local? Like, does it try to match the whole sequence?
It's not. It's more a kind of because it's not as the same as local and global. So it's more for DNA. It's more trying to look for global alignment, but it should be able to tolerate some gaps. So it's kind of seed extension that we do on a local, on a global alignment. So you try to find the best seed position, and you will try to extend on the side. And if you don't find, you will start another thing and try to put together. So it's not as BLAST or other type of general alignment where you do global or local. The Burwell transformer is a bit different, and but it's for DNA, for the DNA aligner, will be close to what we call global alignment. Whereas when you do RNA, it's more close to local alignment because you want to be able to split your read between the exome. So for BWA, it tends to be similar to a global alignment. Yeah.
Sorry, maybe I missed it. Could you please repeat what's the mate position?
The mate position. So
Yeah.
when you do your sequence, you've got your fragment like that. You've got your read one, read two. So if you are looking at information for read one, the mate position is the information of the read two.
So this is just a number to address the position of.
Yeah, of the other read. Okay. So, as I say, alignment is done by aligner. As I say, when you choose your aligner, you make some choice on the parameters. So that the aligner will try to force the alignment based on the parameter you have choose. So the penalties for your gap, for your mismatch, for everything. So the alignment is not perfect. So there's no perfect alignment. So to increase the quality of your information, we need to refine and rework on this alignment. So the first thing we'll do is we will look at indel realignment. So this is a typical example because most of the aligners have more penalty to create indels than to create mismatch. So in some cases, some indels will be missed, and you will see a lot of mismatch in the region. You don't expect so much mismatch. You expect usually to have a SNP every 1 kb. So when you start to see a region with a lot of mismatch, the indel realignment tool will go over this region and realign the reads, try to insert some indels in the region to see if it could improve the read position. Another thing you want to do is to mark your duplicates. So as we see, we are facing some duplicates. So some read pairs that represent the same initial fragment. So this can come from clustering issue. If you have two large clusters in the old Illumina machines, the clusters are not predefined. So in that cases, a big cluster could be split into 2 clusters, and you can generate 2 sequences of the same initial reads. In the new pattern fossil, all the clusters are predefined, but if some cluster positions are empty, one molecule could jump from one cluster to one cluster to the other. So that can make you a different cluster. Another problem is the PCR. When you PCR, if you increase the number of PCR, you can generate several copies of your experiment. This one is not really duplicates, but it's a problem of sister. It's when you two sequences merge together and create a kind of chimera. So what you want to do, you want to only count 1 read per initial DNA fragment. So what we do when you mark duplicates or remove duplicates, you will look at your data and mark all the reads that come from the same initial DNA fragment, and you will take the ones that have the highest base quality in general, to keep the more informative one. Another type of refinement you can do is what we call the base recalibration. So the vendor of the machine can to inflate the value of the base quality they generate, and because these base quality are usually influenced by the position on the reads, as we see the base quality drop at the end, but also by the genomic context. So what we do with the base recalibration, so we take the base quality distribution and it's split, for example, by the genomic context, and you expect in a normal situation that every data should have the same base quality depending on not influenced by the genomic context. So in that case, the recalibration, we recalculate the correct base quality based on all this information.
It's just a question.
Yes.
Last point.
Yeah.
So basically, with this tool, you feed the reference genome and the reads, and based on the genomic context, it will recalibrate the reads.
So the context, the position, the cycle, you take all this information, build a model, and apply this model to all the read to recalculate and recompute the base quality. Because the base quality will be used for during the variant calling, so you want the most confident base quality. So when you have done that, you now have your most highest quality alignment file that you can use for your variant calling. But as I say, what is important is all along the way to take metrics, because it will tell you, it's where, looking at it, when you look at your metrics, you will be able to see if one of these steps didn't work, or is there something particular in your data. So metrics is really important. The different type of metrics, there's a lot of them, so I cannot list everything, but take the most metrics you can find. Most of the time, you will have extra metrics. You will not perhaps look at them, but if there's any issue on your data, having these metrics will really save you sometimes. Many tools provide the metrics. Some tools, BVA tool, GTK, which is now a mix of Picard and GTK for the last version, give really interesting metrics. On my side, if I have 4 metrics to choose, this is the 4 one I will look at. So the metrics of the trimming, how the trimming went, how many reads I lose, how this kind of thing. Alignment metrics, how many reads, percentage of alignment to be able to check for my reference. Metrics on the depth of coverage. So when I design my experiment, I designed for having, for example, 50x of coverage. So did I have 50x in general, and then how the 50x is homogeneous along the genomes. So this is good information about the quality of your experiment. And the inter-size, because when you do your shearing, you target for a size, and if you see that you have completely different inter-size, that probably something went wrong in your library preparation. So metrics, really important. So last part of the workflow for DNA is to do the variant calling. So either for a single nucleotide variation or for a structural variant, but this is what we'll cover in module 4 and 5 tomorrow. So now you have the theory. Now we can pass to the practice and have fun. Thank you. Yes?
If you were using different methods, like not looking at very calling like ChIP-seq or ATAC, is it the same? Would you use the same metrics and still do the recalibration for the base quality where it's maybe not as critical?
For ChIP-seq, you may be able to skip base quality recalibration if you only do peak calling, for example, and motif. But now a lot of people are trying to do SNP calling or SNV calling just to see if there any influence of the SNV on the binding of the proteins. So I would say, if you're really interested only in a peak calling, yes, you can skip it. In terms of indel realignment, you will see that now there's one institute, which is a broad institute that creates the GTK, which provides the GTK best practices, which is what most of people use to follow their different type of analysis, what we used to, and they develop a variant coder that do local realignment when you do the variant coding. So they do not recommend to do index realignment anymore, but me, I would recommend to do it, because if you don't do it, you will be stuck to use only a small number of variant coders that do local realignment at the same step. So if you want to have freedom, still using the index realignment. Yes?
I have just a quick question. So what is the benefit of using a tool like NGMLR for long read alignment versus like BWA?
It's just it's because the error model in long read is different, and so you can use BWA and set up it for long reads, but it will ask you a lot of knowledge to choose the good parameters. Whereas the other aligner, which are already set up to deal with long read with specific error models, will be more easier for you. In terms of benefit, if you are good enough and you have the knowledge to set up BWA correctly, then you could use both.